I welcome you all to yet another interesting series of portion of science presented by IGIB Pulse. So for today's discussion, we have Ms. Ritu Chauhan, a PhD student in Dr. Bhavana Prasher's lab at CSIR-IGIB, and also we have Dr. Pratibhan Rengarajan from AIIMS, New Delhi, to have a very short discussion about their recently published paper, which was entitled Heart Rate Variability During Head-Up Tilt shows inter-individual differences among healthy individuals of extreme Prakriti types, which was recently published in Physiological Reports. So before we start with the discussion, I heartily welcome you for this, for joining us here and about to share your opinions about how was your journey in publishing this paper. So to start with, I would ask you to provide a very brief introduction about the research interest of your lab, or what is the primary focus of your PhD topic is on?
Yeah, thank you, Ms. For the nice introduction. So basically, our lab focuses on explaining inter-individual variability in health as well as in diseases through a unique approach of Ayurgenomics.
Yeah.
Where we use the principal principle of Ayurveda and integrate with them with genomics.
Right.
So actually today's time, precision medicine or individualized treatment is well needed. So we can say even identical twins who grow together with the with same DNA or parents have different personalities. So it can't just be nature or nurture. So when the biology of twins is so different, so that how can one approach be applied to such a heterogeneous population where there are contrasting differences in genetic, ethnicity, or in response to environment. So we can say in terms of therapeutic or preventive approaches. So in Ayurveda, individuals are stratified or segregating into 7 constitution types based on multi-system phenotypes termed as Prakriti. So what is Prakriti? So basically Prakriti is a proportion of 3 doshas. 3 doshas are Vata, Pitta and Kapha. So what is Vata, what is Pitta and what is Kapha? So Vata is a kinetic and is a metabolic, and third one is Kapha is a structural component of our body.
Okay.
So for the first time in 2008, our lab reported genomics and biochemical level variations among healthy individuals in extreme Prakriti types. So further, we have also reported variations at genetic level, metagenome level, and also at cellular level in terms of baseline and in response to UV. So on the basis of this literature, on the basis of these background of our lab, so as I am a physiologist by training, so I asked a question. So we so do Prakriti type shows variations at basal physiological level or in response to physiological stress such as orthostatic stress or head-up tilt. So this was my primary PhD focuses basically.
Okay.
Yeah.
All right. So you have very nicely put on how your lab, what is the scientific domain of your lab and how your work is mostly focused on what are all the explorations you're doing. So that was very well explained, Ritu. So I have also gone through your paper, and I got to know that you had mentioned how you use the heart rate variability among the individuals to identify the autonomic dysfunctions. So I would like to ask you, like, how did you end up in such an idea or a hypothesis, or how did you process this approach initially?
Yeah. So as I mentioned that I wanted to see Prakriti specific variations at physiological level using orthostatic stress. So first I want to talk about more on orthostatic stress basically. So as we all know, the postural changes. The postural changes are an important aspect of our daily life. So which has an enormous influences on our physical and well-being. So in sudden upright posture, if our compensatory reflex system activation is impaired, this may lead to hypotension, like low BP, or tachycardia, like high heart rate, or even in the extreme cases, it may lead to syncope.
Okay.
So even recently, it has also been studied that the orthostatic intolerance occurred after COVID infection. So it is very important to study this kind of stress that hamper our daily life. So and also in the literature, it is reported that that even healthy individuals respond differently towards this kind of stress. So the identification of predisposed group or individuals to autonomic dysfunction related disorders in healthy state is a major challenge. So in this study, we exposed healthy individuals of different Prakriti types to head-up tilt or orthostatic stress and measured continuous heart rate variability. So heart rate variability is used to assess our autonomic nervous system activities. It is a non-invasive technique to assess the autonomic nervous system activities. So for this, we have collected data from two cohorts. First one is Vadu cohort from Pune, and second one is Delhi NCR cohort, a cohort Delhi NCR people with Delhi NCR. So data from Vadu cohort was collected as a part of Ayurgenomics consortium, and the data from Delhi NCR cohort was collected by Dr. Pratibhan, as we have Dr. Pratibhan we have. So from AIMS during his PhD. So Dr. Pratibhan will elaborate further on this work.
Okay. Yeah, sure, sir.
Sure. This is based on my part of my PhD work. Actually, we wanted to collect some more parameters also we have collected. So this is only a few parameters. So basically, our my AIMS lab focuses on the physiological function test. So we have a clinical autonomic function test lab. So we do autonomic function tests regularly for patients, and also we have vascular functions also. So basically, our lab focuses on the clinical, autonomic, and vascular function. So this, so our question is that we see not many patients daily for the clinical autonomic function testing, and we see differences in the clinical subject, in the patients itself. So we don't know how the healthy individuals will vary in autonomic function test. So only we need a framework. That framework is given by the Ayurveda Prakriti features, like Prakriti phenomena. So it allows to segregate the healthy individuals. That's the main thing in the Prakriti. So we classified the healthy individuals based on Prakriti, and we did the autonomic function test. So first we did the baseline HRV, and we couldn't find any changes in the baseline HRV. And we used the stress test. Like we tilted the tilt table 260°, and we found what are the possible changes we observed in the different Prakriti features. That is about this study.
Okay. All right. So I think you both gave a very clear picture of how you ended up with the idea and how you executed with the approach. So with this, on the same note, my next question will be like any task which we undertake will be there with some challenges in between. So I would like to ask from you, like what kind of challenges you had undergone during the course of this work, either be in terms of experiments or kind of situation with pandemic coming in between, like what were the challenges you had to go through?
So Mercy, the main challenges, the main challenge basically was the recruitment of the healthy individuals. So, and we have taken extreme Prakriti individuals in this study, and the extreme Prakriti individuals is only 8 to 10% of the total population, of a population basically.
Okay.
So to achieve a statistically validated sample number, a massive level of screening was required.
Right.
So to recruit the extreme Prakriti individuals.
Right.
And after that, convincing participants to means HRV assessment is another challenge. So during HRV, we need to ensure a proper ambient environment for each and every individuals to relax and capture their baseline HRV.
True.
Because this is important to see the response to stress from the baseline of each individual. So and also we have extensive protocol, and each step so each step is crucial to capture accurate data for the implementation. So this is another challenge. Next challenge we faced during the manuscript writing. So this was the first report on HRV variations among healthy individuals. So there was no literature found to discuss our findings basically. So HRV literature are available. So on the basis of in BMI HRV variations, in age-wise HRV variations, in various diseases HRV variations was reported basically in the literature, but no study was found to on healthy individuals. So HRV variation for healthy individuals. So this remain a major challenge for our team.
Yeah, I think you got up. You came up with one of very unique challenges. Like especially in writing a manuscript, you didn't have a background literature already available. You had to dig up everything by yourself. That was kind of a very true to yourself, whatever you had gone through while working on this paper. And next I would like to ask, like we all know or we all believe that success comes through hard work and toil. So the reaching the destiny of to the stage of publishing your paper. So how was it? Was it a one-shot submission, or you had faced any rejections? If by any chance you had faced any rejections, how did you exactly tackle it? Was it like, obviously it would have been sad. I would like to hear your experiences on that.
So Pratibhan sir will answer this question.
Okay. Yeah, sir, sure.
Okay. First we submitted to the journals. So they given some comments and they rejected. So they what was their understanding was that they suggested why you are taking Prakriti as a classification phenomena. So why can't you classify the humans based on BMI or any other parameter? So they didn't understand the importance of Prakriti feature. So in Ayurveda, they have described that Prakriti is both anatomical, physiological, psychological, and all the characteristics are there in the Prakriti features itself. So it is a multi-system phenotype characterization. So that is the Prakriti. So they thought it is like a single phenomena, like it is like a single factor that will classify the humans, like based on the BMI. We can keep a cutoff value and we can classify the humans. Likewise, they considered as a Prakriti as one of the classification pattern.
Yeah.
So that was their first comment. So after that, they suggested the results are not convincingly explaining the physiological parameters. That was their second comment. So we submit, after that we submitted another journal, we tried another journal. In this recent physiological reports, they suggested they have given a very good suggestion. So they accepted this Prakriti feature and everything, and they suggested that you have taken extreme Prakriti people. Why can't you do this study mixed Prakriti people? That was their suggestion. So again we did the same analysis in the mixed Prakriti people also. We had the data. So based on their suggestion, it was actually we got a very good result even in the mixed Prakriti population also.
Okay. Actually, that was I had that as my next question. Like, if you had faced any rejections, like what kind of reviewers' comments? Like, was it reasonable, or because we get to see that there are some comments from reviewers which are like unanswerable, or it is not pertaining to what research we are doing. So like you had said, the very first paper where you had submitted, you got some kind of why kind of comments, whereas the second paper, you mean to say that they gave very good comments which also enhance the quality of your paper. So yeah, that was indeed a very good point, sir. So this brings me to the end of our discussion here. So you had explained how you started with the idea, how you made the approach to work on this idea, and how was your journey towards the submission, and how you faced with rejection, comments, and everything. So throughout this whole process, it can be either for you or for Ritu, like what was the learning, be it to work on the experiments, or what is that one learning you had acquired and you would like to share with your fellow colleagues who are pursuing a PhD to like follow it, some kind of what you personally experienced which you would like to share with friends who are also working on a PhD right now.
Yeah, so the suggestion from my side is to overcome your fear of paper writing.
Okay.
So there is always a first time for everything. So
Yes.
you will find yourself to go blank in the beginning, but subsequently when you write all observations and results, you will get a motivation to complete the manuscript. So manuscript even as soon as possible. So believe me, first paper acceptance gives true happiness that you have achieved something significant, and also keeps you going further with more zeal and enthusiasm. So Dr. Pratibhan, would you like to add something into this?
Yes, sir.
So what in this study, so the difficult what we found is in the subject recruitment only basically.
Yeah.
So convincing healthy individual is the very difficult because they are healthy. They tell that we don't have any problem and we are healthy, so we don't have time. That's their basic. So that we have to convince and overcome. That is first thing. And second is that don't be rejected based on the reviewers' comment. So
Right.
there are some good reviewers are available. There are some good editors are also available. So surely they will accept our paper. So they will see, they will think from our point of view, not from their point of view. So we'll find some good journals are available still.
Okay, that was really a very interesting and very insightful discussion with you both. And lastly, I would also like to congratulate both of you for such a wonderful paper. And as you had mentioned, a very unique one from your domain also, for the first time, a unique report from your domain also. Thanks a lot for sparing your time and adding your opinions and how you came to this journey to us. Thanks a lot for joining. Thank you, Ritu. Thank you so much.
Thank you.